This video demonstrates the process of sectioning formalin-fixed paraffin-embedded tissue for histological and immunohistochemical analysis. Thin sections of the tissue are cut, which can be stained and imaged to visualize protein expression. Freshly processed and embedded blocks will have a layer of wax covering the tissue. Before sectioning, use the microtome to trim away excess wax at room temperature. To prepare the microtome, carefully remove the blade from the case and insert into the holder. Lock the blade in place and set the blade clearance angle and section thickness to 12 micrometers for trimming. Insert the block into the block holder, making sure the microtome handle is locked. Start trimming the block by turning the microtome wheel. Ensure the tissue in the block is fully exposed at the surface. This process will also align blocks with your microtome blade, ready for sectioning. Once trimmed, place the paraffin-embedded tissue blocks on ice to cool to make sectioning easier. Whilst the blocks are chilling, warm a water bath containing distilled water to just below the melting point of the wax used, roughly 40 degrees Celsius, and clear off any debris using a Kimtech wipe. Place the cool block back into the block holder and bring the block close to the blade. Set the section thickness to 5 micrometers. This is recommended as this is approximately the thickness of one cell. Turn the microtome wheel at a steady pace. Use chilled forceps to grab hold of the first section and gently lift the ribbon as it extends from the blade with each cut. You can also gently blow down on the blade as you cut to help flatten the sections. Tissue sections are transferred to the water bath and wrinkles allowed to disappear. Sections can be separated using a pair of chilled forceps. Allow wrinkles to disappear before transferring the sections into charged slides, avoiding air bubbles. Transfer slides to a slide holder. Finally, allow the slides to dry in an oven at 50 to 55 degrees Celsius for 4 hours, or 37 degrees Celsius for 8 hours, and store slides at room temperature until ready for use. If the ribbon is not forming, the paraffin wax might be too hard. You may need to use paraffin with a lower melting point or adjust the room temperature. Knife lines running through your tissue sections are caused by dull or scratched blades. Replace the blade and ensure the knife tilt angle is properly adjusted for the tissue. Thicker sections are more likely to form wrinkles. Floating the sections in a water bath should remove any wrinkles. If the temperature is too cold, this can prevent sections from fully flattening. Tissue with high lipid content, such as brain and breast tissue, can explode when placed into the water bath. This can also happen if tissue has been poorly processed. To avoid this, try lowering the temperature of your water bath. Alternatively, you can add a drop of dish soap or alcohol in the water bath to break surface tension. However, make sure to account for this addition in downstream assays.